Hello, my name is Natalie Verkade and this is my presentation on Grehlin. In its active form, Grehlin has been identified as a 28 amino acid peptide. Most of it is secreted by specialized cells in the stomach, enteroendocrine cells, and these are scattered in the gastrointestinal tract mucosa. Most of the remaining secretion originates in the small intestine, although there are some tissues outside the GI tract that have been shown to secrete Grehlin. Grehlin is known as the hunger hormone as it's secreted in response to fasting and acts as a strong appetite stimulant. Levels increase prior to eating and decrease once you are in a fed state in a normal, healthy individual. Grehlin acts both locally and in the hypothalamus. In the stomach, Grehlin acts as a paracrine hormone that modulates the action of gastrointestinal hormones controlling gastric emptying, motility, and energy balance. Grehlin regulates food intake at a central level by exerting its effect on receptors in the hypothalamus implicated in appetite control. The maintenance of energy homeostasis is dependent on the balance between energy intake and energy expenditure. So together with leptin acting as the antagonist, which is released from the adipose tissue, Grehlin increases appetite and stimulates appetite. Once in the fed state, leptin is secreted by the adipose tissue and it removes the stimulus for eating. Interestingly, the receptors for both leptin and Grehlin are the same in the hypothalamus. Once Grehlin is synthesized, mostly in the stomach as a peptide hormone, it is transported in the blood dissolved in plasma. On reaching the target cells, it binds to a G protein coupled receptor. This directly activates the amplifier enzyme phospholipase C. This in turn activates second messenger systems of IP3 to release intracellular stores of calcium and DAG to increase phosphorylation of AMP-activated protein kinase. Together, the phosphorylation and the increase of calcium produces the cellular response. As well as being responsible for stimulating appetite, Grehlin is known to encourage the secretion of growth hormone, which is quite independent from its appetite stimulating properties. It encourages the release of growth hormone releasing hormone, which releases growth hormone. During periods of fasting, this has a number of functions, including the synthesis of proteins to allow tissue growth. It mobilizes fats from adipose tissue. It also has effects on glucose homeostasis within the body. In the liver, it promotes glycogen breakdown to increase blood glucose levels. Growth hormone levels are the highest after the first few hours sleep. As Grehlin levels start to increase 5 to 6 hours after eating, it is likely that this stimulates the growth hormone cascade by stimulating the release of growth hormone releasing hormone from the hypothalamus. Grehlin also interacts with the hormones secreted by the pancreas, most particularly insulin. Plasma Grehlin levels increase during fasting, and this decreases glucose uptake as it inhibits insulin and therefore promotes glucose sparing for the tissues. In obese individuals, there is a reduction in circulating Grehlin levels, which may explain why they have an excess of circulating insulin, which is often mistaken for diabetes. Prader-Willi syndrome is perhaps one of the best known syndromes connected with excessive Grehlin secretion. It's a rare genetic order that it appears from birth. It begins off with infantile hypotonia and developmental delay and can lead to mental retardation and behavioral disorders. It tends to lead to a high level of obesity and presents itself between 1 to 6 years of age. Sufferers of Prader-Willi syndrome have very abnormal food-seeking behaviour. They hoard food and they also eat spoiled or rotten food. They steal food. They're known to have to put locks on pantries and refrigerators. It is thought that excessive secretion of Grehlin plays a role in this syndrome, as there is no off switch in that the levels of ghrelin do not decrease after feeding. Therefore, syndrome sufferers are always hungry and are always seeking food. Further studies have shown that appetite-stimulating properties of ghrelin are separate and independent from the secretion of growth hormone, as Prader-Willi syndrome sufferers often have short stature. However, the excessive secretion of ghrelin and the fact that it doesn't decrease after meals leads to the overeating, and this is consistent with the idea that ghrelin is a physiological regulator of appetite in humans, and it is independent of its secretory role, stimulating role in the secretion of growth hormone. In Prader-Willi syndrome, the physiological effects are quite apparent. Excessive ghrelin secretion leads to an appetite which is disproportionate to normal levels. Ghrelin levels do not fall after a meal, so Prader-Willi syndrome sufferers are always hungry and seeking food. Current treatment is the human growth hormone, and this further supports ghrelin's role in stimulating appetite is independent of its stimulation of growth hormone secretion. The role of ghrelin in Prader-Willi syndrome has not yet been firmly established, although it is apparent that plasma levels of ghrelin are generally elevated in Prader-Willi syndrome sufferers.